In section 48.1, we discuss how a neuron's organization and structure enable it to transfer information. One example of this includes the squid. The squid surveys its environment using its image-forming eyes. When it finally spots prey, signals traveling from its brain to neurons in its mantle causes muscle contractions that propel the squid forward. The squid's hunting activity illustrates the three stages in information processing, such as sensory input, integration, and motor output. Sensory neurons transmit information from eyes and other sensors that detect external stimuli, such as light, sound, touch, heat, smell, and taste, or internal conditions, such as blood pressure, blood carbon dioxide level, and muscle tension. This information is sent to the brain or the ganglia. Neurons in the brain or ganglia integrate the sensory input, taking into account the immediate context and the animal's experience. The vast majority of neurons in the brain are interneurons, which only make local connections. Motor output relies on neurons that extend out of the processing centers in bundles called nerves to generate output by triggering muscle or gland activity. For example, motor neurons transmit signals to muscle cells, causing them to contract. The neurons that carry out integration are organized in a central nervous system, or CNS, which includes the brain and the longitudinal nerve cord. The nerve cells that carry information into and out of the CNS constitute the peripheral nervous system. As we see in the next diagram here, there is a sensor, which is the ear, which brings sensory input into the brain through integration. The brain then processes this information and then enables the motor output through the effector. The central nervous system and the peripheral nervous system work together hand in hand in this process, as you can see here. Next, we move on to neuron structure and function. The ability of a neuron to receive and transfer information is based on a highly specialized cellular organization. Most of a neuron's organelles, including its nucleus, are located in the cell body. A typical neuron has numerous dendrites, which are highly branched extensions that receive signals from other neurons. A neuron also has a single axon, which is an extension that transmits signals to other cells. Axons are often much longer than dendrites, and some, such as those that reach from the spinal cord of a giraffe to the muscle cells in its feet, are over a meter long. The cone-shaped region of an axon, where it joins the cell body, is called the axon Hillock. As we will see, this is typically the region which signals are able to travel down the axon and are generated. Near its end, an axon usually divides into several branches. Each branched end of an axon transmits information to another cell at a junction called the synapse. This part of each axon branch that forms the specialized junction is a synaptic terminal. At most synapses, chemical messengers called neurotransmitters pass information from the transmitting neuron to the receiving cell. In describing a synapse, we refer to the transmitting neuron as a presynaptic cell, and the neuron, muscle, or gland cell that receives the signal is the postsynaptic cell. Depending on the number of synapses a neuron has with other cells, its shape can vary. To function normally, the neurons of vertebrates and most invertebrates require supporting cells called glial cells, or glia.
The next two drawings display the basis of the membrane potential. In the first drawing, the values shown represent the approximate concentrations in millimoles per liter for ions in the fluids within and surrounding a mammalian neuron. K plus is the potassium concentration, Na plus the sodium concentration, Cl minus the chloride concentration, and A minus represents the other anions. In this next drawing, the small yellow squares represent Na+, the small red squares represent K+, the orange pump is a sodium-potassium pump, the dark purple channel is a potassium channel, and the light purple channel is a sodium channel. So in this drawing, the sodium-potassium pump generates and maintains the ionic gradients of Na+ and K+, as shown. The pump uses ATP to actively transport Na+ out of the cell and K+ into the cell. Although there is a substantial concentration gradient of sodium across the membrane, very little net diffusion of Na+ occurs because there are very few open sodium channels. In contrast, the large number of open potassium channels allow a significant net outflow of K+. Because the membrane is only weakly permeable to chloride and other anions, this outflow of K+ results in a net negative charge inside the cell. The next two drawings model a mammalian neuron. Each container is divided into two chambers by an artificial membrane. Ion channels allow free diffusion for particular ions, resulting in the net flow represented by arrows. In the first drawing, the presence of open potassium channels makes the membrane selectively permeable to K+, and the inner chamber contains a 28-fold higher concentration of K+ than the outer chamber at equilibrium. The inside of the chamber is negative 90 MV relative to the outside. In the second drawing, the membrane is selectively permeable to Na+, and the inner chamber contains a 10-fold lower concentration of Na+ than the outer chamber. At equilibrium, the inside of the chamber is 62 MV relative to the outside.
In section 48.4, we discuss how neurons are able to communicate with other cells at synapses. Some synapses, called electrical synapses, contain gap junctions, which do allow electrical currents to flow directly from one neuron to another. Both vertebrates and invertebrates use electrical synapses to synchronize the activity of neurons responsible for certain rapid, unvarying behaviors. Going back to the squid example, electrical synapses associated with the giant axons in squids and lobsters facilitate the swift execution of escape responses. There are also many electrical synapses in the vertebrate brain. However, the majority of synapses are the chemical synapses, which enable the release of a chemical neurotransmitter by the presynaptic neuron. The cell body and dendrites of one postsynaptic neuron may receive inputs from chemical synapses with hundreds or even thousands of synaptic terminals. At each terminal, the presynaptic neuron synthesizes the neurotransmitter and packages it in multiple membrane-bounding compartments called synaptic vesicles. The arrival of an action potential at a synaptic terminal depolarizes the plasma membrane, opening voltage-gated channels that allow calcium to diffuse into the terminal. The resulting rise in calcium concentration in the terminal causes some of the synaptic vesicles to fuse with the terminal membrane, releasing the neurotransmitter. The neurotransmitter then diffuses across the synaptic cleft, the narrow gap that separates the presynaptic neuron from the postsynaptic cell. Information transfer is much more readily modified at chemical synapses than electrical ones. There are a variety of factors that can affect the amount of neurotransmitters that are released or the responsiveness of the postsynaptic cell. These modifications underlie an animal's ability to alter its behavior in response to change and form the basis for learning and memory. Next, we look at this diagram of a chemical synapse. In step 1, this is where an action potential depolarizes the plasma membrane of the synaptic terminal, and then forms step 2, which opens the voltage-gated calcium channels in the membrane, triggering an influx of Ca2+. In step 3, the elevated Ca2+ concentration in the terminal causes synaptic vesicles to fuse with the presynaptic membrane. In step 4, the vesicles release the neurotransmitter into the synaptic cleft. In step 5, the neurotransmitter binds to the receptor portion of the ligand-gated ion channels in the postsynaptic membrane, which open the channels. In this synapse illustrated in the diagram, both sodium and potassium can diffuse through the channels. In step 6, the neurotransmitter is released from the receptors, and the channels close. The synaptic transmission ends when the neurotransmitter diffuses out of the synaptic cleft, is taken up by the synaptic terminal or by another cell, or is degraded by an enzyme.
Because depolarizations bring the membrane potential toward threshold, they're called excitatory postsynaptic potentials, or EPSPs. Hyperpolarizations are called inhibitory postsynaptic potentials, or IPSPs, because they move the membrane potential farther from the threshold. A single EPSP is usually too small to trigger an action potential in a postsynaptic neuron. During temporal summation, two EPSPs occur at a single synapse in such rapid succession that the postsynaptic neuron's membrane potential has not returned to the resting potential before the arrival of the second EPSP. In spatial summation, EPSPs are produced nearly simultaneously by different synapses on the same postsynaptic neuron. Two or more IPSPs occurring nearly simultaneously have a larger hyperpolarizing effect than a single IPSP. Through summation, an IPSP can also counter the effect of an EPSP. Acetylcholine is one of the most common neurotransmitters. It's excitatory to vertebrae skeletal muscles but inhibitory at other sites. Norepinephrine acts as a hormone, while dopamine and serotonin influence sleep, mood, attention, and learning. While GABA and glycine are inhibitory, glutamate is excitatory. Substance P, which is a neuropeptide, is involved with perception of pain. Nitric oxide releases dissolved gases and work like hormones.
The first step of the conduction of an action potential is seen as the action potential is generated as sodium flows inward across the membrane at one location. The second step begins as the depolarization of the action potential spreads to the neighboring region of the membrane, reinitiating the action potential there. To the left of this region, the membrane is repolarizing as potassium flows outward. The third and final step occurs as the depolarization and repolarization process is repeated in the next region of the membrane. In this way, local currents of ions across the plasma membrane cause the action potential to be propagated along the length of the axon. This first graph shows graded hyperpolarizations produced by two stimuli that increase membrane permeability to K+. It can be seen that the larger stimulus produces a larger hyperpolarization. The second graph shows graded depolarizations produced by two stimuli that increase membrane permeability to Na+. The larger stimulus produces a larger depolarization. The last graph shows the action potential triggered by a depolarization that reaches the threshold.